Hello, and thank you for joining us. I am Jasmine Navarez, Digital Content Coordinator at Simulations Plus. I'll be hosting today's webinar behind the scenes, making sure we don't have any technical difficulties. In this webinar, Crystal Pharmatech co-founder and Chief Business Officer Robert Wenslow will discuss optimizing benchwork and computational approaches to assess CMC. Our Business Development Director, Andrew Mueller, will serve as our moderator today. A few housekeeping items before we begin. We take your privacy rights seriously. By registering for and attending this event or participating in the Q&A session, you are allowing us to contact you for follow-up. You may ask questions via the questions panel on your dashboard at any time. If you need assistance, please use the hand raise icon. We'll address questions in the Q&A during and after the presentation. And then I'll go ahead and pass it over to Andy.
Yes, thank you very much, Jasmine, for getting us started. Thank you to our participants for carving some time out of your day today. And thank you very much to our friend and presenter, Bob Wenslow. A little introduction to Bob. After an illustrious 15-year career at Merck and Rahway, Bob co-founded Crystal Pharmatech, where his interests included and continue to include API formulation, selection, and assessment. Bob remains currently obsessed with bringing in silico tools, such as PBPK modeling and GastroPlus, for example, to the pharmaceutical formulation development masses. In this webinar, Bob will dazzle you by outlining some advances, thoughts, and optimized workflows he and his colleagues at Crystal Pharmatech have used to assess small molecule CMC developability with very limited material. Thanks, Bob. Please take it away.
Thank you, Andy. And illustrious is a stretch, number one. Dazzle is another stretch. So we'll see where we go. And for those 12% of the people that don't like animals after this, we'll make sure that you do. Also, before I get started, this is not all of my doing. I'd like to thank everyone on my team, all of the collaborators that we've worked with in the polymorph prediction, the GastroPlus, Simulation Plus, excellent partners. And also specifically, I'd like to give a shout out to Paul Lunar, who specifically helped me with some of the slides that we have here. And so what are we trying to do? And so what we want to try to do is use what's the best use of small amounts of material early on in your development program to rapidly assess the risk for form and formulation and get the most bang for your buck on every experiment and specifically every PK study. We want every experiment to be data rich, no wasted work, and use industry standards and really have a smart form and formulation development. And while we'll outline the benchwork, we also want to take advantage of in silico technologies that are well established. So all of the technologies that we're going to be talking about are being used on a daily basis by most larger innovative pharmaceutical organizations, but they all have different approaches. And so it's truly an unbaked cake. And so I'm going to be outlining some of the tools that we use, but how specifically they'll be used in your program is really going to vary on what your program is, your target product profile, and how do you want to move forward in the clinic. And as I mentioned, most or all large pharmaceutical organizations are using this technology on a daily basis as part of their risk assessment. And we really want to bring it to the masses and in a cost-effective fashion. So we really want to not have the front loading, and we want you to see this as saving you money instead of actually costing you for your development. And so in lead optimization, right around that candidate selection stage, some of the challenges that we might have, especially in late lead op, is small amounts of material from medicinal chemistry batches, probably not fully characterized. And we would really like to maximize the amount of information that we're getting from that small amount of material, and using in silico tools as much as possible to make data-driven decisions to minimize late stage failures, or specifically PK study failures, GLP tox failures, and first in human failures for exposures. And so understanding the risks of the form and formulation of your compound to minimize those late stage failures. So what are we talking about? And we're talking about combining in silico modeling, in vitro testing, and predictive analysis to give you a data-driven candidate slash form formulation selection going into phase 1. And so early ADME toxicology screening and development, we can predict ADME profiles, regional GI absorption, metabolic pathways, potential toxicities, and this is without any material. This is basically with a two-dimensional structure, basically a SMILES, and using predictive software like GastroPlus through Simulation Plus. We can also do, with very small amounts of material and very minimal amounts of solubility information, take use of industry benchmarking. And what do I mean by that is, say a company like Merck or Pfizer is developing, hundreds of compounds a year, and they have these massive databases. And so based on certain solubility properties or certain physical properties of the compound, how does that move you in your development pathway? And specifically, we'll be talking about developability classification, dose number, dissolution number. And one of the major advancements in the last few years has been really the ability of polymorph prediction, being able to predict crystal forms and direct us towards isolating those crystal forms that are predicted. And it's really not as expensive or time consuming as you had seen in the past. It's really getting to the point where we're bringing it down to the masses, especially with our partner Biosimulytics. So what can you predict without with very small amounts of material and a two-dimensional structure? Specifically with the GastroPlus, you can predict pKa, log P, permeability. You can solubility. Now, the solubility point is no one's doing a great job of predicting solubility. That's one of the big concerns. But with just very small amounts of material, less than 25 milligrams, we can find a crystal form, not necessarily the best crystal form, but get a crystal form solubility, and also an amorphous solubility with some of the techniques that I'll be showing, and get an understanding of the advantages of using an amorphous form over a crystalline form. We can get ADME prediction, human PK prediction. Again, we don't like to use the GastroPlus software just straight in silico. What I'll show is with getting just small amounts of characterization data from your compound, inputting those accurate data like pKa, like solubility, and also in vitro metabolic data, we get a much more accurate GastroPlus PK model. And you are going to have some PK studies, in vivo studies. So fitting that data as well allows us to have a much better prediction. So the benchmarking, we'll be talking about, again, amorphous solubility and preclinical dose number, dissolution number. And from a polymorph prediction standpoint, what is your most stable crystal form? And so very early on, with say 10, 15 milligrams of material, you can grow a single crystal. And having that crystal structure, we can then lean on that in the polymorph prediction and get an understanding of where we are in the polymorph landscape. So the form that we are making currently, is that close to the lowest energy? Is it the lowest energy? Does it look like there's going to be a ton of polymorphs? And again, early on, if we're in the candidate selection stage, we're just trying to rank order the compounds and seeing, okay, which one is going to have, significant amounts of polymorphism versus one that's going to have really one low energy stable form that's going to be easily accessible. More importantly, which compounds are not going to crystallize. And so we're working with biosimulatics as well to find out which compounds are not going to crystallize. So the sooner you can find those problem cases, the faster you can use that in your assessment of what compounds to move forward. Again, in vivo is going to be driving most of this decision, but you want an understanding from a formulation perspective of a rank order of your compounds to help make the best candidate selection. We also use a software called Cosmologic. And again, these aren't the only tools out there. This is just some examples that we work with specifically to get relative solubility. It works best when you have at least one accurate solubility value, but then you can go in and predict solubility in binary, ternary solvent systems, changing water activity. We also use Cosmologic a lot for co-crystal formation and trying to find the best synthons, the best co-formers to use in a screening. And it's actually not bad. It does a pretty decent job of rank ordering some of the co-crystal formers, and at least telling you if it makes sense to do a co-crystal screening. And more recently, we've been implementing it to crystallize new forms. And so you get this polymorph, predicted polymorph landscape. How are you going to crystallize those forms? And Cosmologic can actually give us some insight into that. So what is this tier one? What are these tools? So with 25 to 50 milligrams of material, the first thing we want to do is to get something that's crystalline. We want an understanding of the amorphous phase, which you can quickly get with a modulated DSC, getting a glass transition, and then seeing if you could even do a polymorph experiment at high heating above the glass transition temperature to see if it's going to crystallize. First and foremost, with your batches, we want to know what they are, right? Are they crystalline? Are they amorphous? So get a quick X-ray, non-destructive, 2 to 5 milligrams of material. Is it highly crystalline? Is it amorphous? Is it somewhere in the middle? Get a DSC, but don't just do a standard DSC. Do some cycle experiments. Get a modulated DSC. Get the most bang for your buck. Getting a glass transition temperature, a melting point. You can even use those values from the DSC to predict an amorphous solubility. But we're actually going to get an amorphous solubility with the small amounts of amorphous material using pion systems. The pion microdis system will allow us to do amorphous solubility determination with, say, 5 to 6 milligrams of material. And it's a live kinetic measurement of the amorphous solubility and the precipitation rate of the amorphous. It's difficult to pull the sample out for an X-ray, but you do measure concentrations very well using that system. One of the things I'm going to have on my grave is, do you have a PLM for that particle? So as I was walking this morning, I realized that Darwin had a microscope way back in the day, and we don't use ours nearly enough. So material that comes in, get a quick microscope image. You could see nice crystals, maybe not nice crystals, amorphous material. It's a really powerful tool, especially with the hot stage. You can monitor what happens upon heating. You're going through an actual melting point. what are those bumps on the DSC actually, what's actually happening there? Well, you can visually get a representation of that with your hot stage microscopy. And again, TGA, DSC, the ability to get melting points, glass transition temperature, giving an indication of if the material is a solvate, a hydrate, or anhydrous. And the one big piece of data we want to get is FASF solubility, and a highly accurate FASF solubility. This is with a crystalline and an amorphous material. But it doesn't have to be the best crystal. So I'm not saying I'm going to do a polymorph screening with 25 milligrams of material. What I'm saying is we can do a polymorph prediction with no material. And with the 25 milligrams, 80% of the time, we're going to get some type of crystal. And with combining those two, you can then see, well, the crystal that I found, am I at the end already, or do I got a lot of way to go? And so with that crystalline material, we want to get a highly accurate FASF solubility, because this is a worst case from an oral absorption perspective. In vitro is mimicking the fasted upper GI, where most of the compounds are going to absorb. And so understanding that solubility will give you a worst case scenario of how your compound is going to orally absorb. And if you have the amorphous compared to that, then you can get the potential advantage of using the amorphous over the crystalline. And then you start using the Gastro Plus to start understanding what your PK profiles would be with an amorphous material compared to a crystalline material. When you start having a little more material, then things get a little bit better, and you can then start getting accurate log P, pKa. Now, what we've noticed is the Gastro Plus it does a really good job of log P prediction. pKa better than anything else predictive that we have, but what we've still noticed with a lot of resonance structures is getting a highly accurate pKa can be tricky sometimes, but it is very important for your development program. And so we can use the Pion system here, Sirius T3, when it's working, to with about 10 milligrams of material to get accurate pKa or log P. Then we can start doing some force degradation. And we're not talking 14-day force deg. We're talking, is your API gonna, blow apart with acid or base or oxidative or hydrolysis? So like a 1-day force deg, start, playing around with the HPLC method, starting to look at what impurities are coming in. From our end, we're not going to be doing necessarily mass spec and figuring out the structure of those impurities, but we do want an understanding of how your material is going to degrade, and maybe what solvents you want to avoid in your crystallization processes. And I don't want to overemphasize, haven't emphasized it, I'll emphasize it, the getting a single crystal, especially now with micro-ED. With micro-ED, your particle size can be very, very small. I don't know the exact micrometers that they're saying they can get down to, but it's being very successful. And so you don't really have to grow these massive crystals anymore. And getting the single crystal from single crystal X-ray diffractometer is not that significant of an experiment. Not trying to minimize it, but with, say, 10 to 15 milligrams of material, you have a pretty good probability of getting something to crystallize large enough either to get the single crystal with the X-ray or with the micro-ED. And with that, you, number one, have absolute, well, depending on how heavy anatomy you have in there, you can get absolute structure determination from absolute stereochemistry, definitely absolute structure determination. But then also you can use that in the polymorph prediction to then really quickly get an idea of where you're at in the polymorph landscape. It really cuts down on the cost and the speed of the polymorph prediction. And I've shown here, again, I don't want to go into this too much, but every compound is going to be different, or every program is going to be different, because your target product profile and your clinical strategy is going to be different. And so that's why having these cross-functional teams all together on the same page of the development is critical from start one. And one of the main points I like to point out here is, stop thinking of salt selection and formulation development as checkboxes, right? You need to start thinking about those. Not that, even if you're not doing a lot of work up front, you need to start thinking about that as soon as you have an idea of your target product profile and what you're thinking about for clinic. Maybe you just want to rapidly go in with compounding in the clinic, and that might be great for what you're looking to do in your clinical strategy, but you need to understand that you're going to eventually have to do formulation development at some point for solid oral dosages we're talking about here. But getting all those cross-functional team members involved early on is critical for the rapid success of a program. But looking at some different tiers here, we want to get, as I mentioned, that baseline characterization. We want to get a crystal. We want to get FASF solubility. We want, if we have a pKa that's amenable to make a salt, even better. We're not going to do a full salt screen, but let's make a salt. Like, let's say we have a pKa of 5, right? Let's make a salt, see what its potential solubility advantages are in FASF, see how rapidly it disproportionates, maybe even get a pH max on the salt form, and really start getting an understanding of where you are in the developability classification and how difficult it is going to be to formulate to get the dosage you need for your clinical studies. So as part of the PBPK modeling, as I mentioned, we really like to use this, and we're promoting our clients to use this as soon as you start having some in vivo PK data with some small amounts of accurate solubility information, log P prediction, pKa information, any understanding of metabolism, which you usually have in, even in like late optimization space, and you'll have permeability, usually like KCO2, and get an understanding of efflux and metabolism pathways, et cetera. And we'll start by fitting the IV of one animal to start getting an idea of how well the clearance is predicting. Then we'll go to the PO for that animal and see where the gaps are, right? See how good this model is fitting and where the gaps are. And then you go to a second animal and start fitting the second animal, and it gives you confidence that your metabolism pathways are understood, your clearance is understood, and then then start working your way up to the PK, the human PK prediction. But what we're really looking at from a formulation standpoint is how is this compound orally absorbed? Is it rapidly dissolving, then precipitating, and then redissolving? Formulation for the most part is really about dissolving and absorbing the compound. And so understanding this is critical to understanding your formulation path. I can't say, pretty much every day or every other day, someone will come to me and say, we need a salt. And you start walking through the questions and realizing that we really don't understand why we need a salt. And are you making that proper assessment? Like, have you fully vetted your free form? Do you know that a salt's going to give you anything, or is it just going to just precipitate right out because of the pKa and the pH max of the compound? Are you going to be in that? Do you really need to be in that amorphous path and don't even mess around with the salt form? And so this PBPK modeling is amazing in helping us understand how the compound is traversing through the GI tract and how it's absorbing. And what it allows us to do is parameter sensitivity. And so once we have a good model, then we can see, okay, if my solubility changes by 30%, what's going to happen to my oral absorption? And this is something, when combined with polymorph prediction, is amazing. And there's a recent article out from the Genentech team on how they used this GastroPlus with the parameter sensitivity, how they used Tiny Tim and the polymorph prediction to say, there's another form out there. We know there's another form out there. We just haven't crystallized it yet. What's the impact going to be? Since they had such a good PBPK model, they were able to tell the impact of the oral absorption if that polymorph actually came out. Well, 7 campaigns later, it came out, right? But they knew what the impact was going to be, and they knew what their plan would be when it actually did indeed come out. The key is, how could we have crystallized that faster? And that's what we're working on with biosimulitics and also cosmologic, the ability to get one of those forms to crystallize. So what is this benchmarking industry standard stuff, right? So literally what we're looking at is accurate pKa, Keiko-2 permeability, which will then predict a human permeability using GastroPlus, and a FASF solubility. With different, if you want to go in with different forms, that's fine. You definitely want to try the crystalline free form, maybe a salt form, maybe the amorphous form. But again, it's not going to take very long to make one salt and just a rapid, either get an amorphous solubility or even make one dispersion, small scale dispersion. And then we start benchmarking the compound against industry standards. So these are all out in the literature. We just gathered them together and put them into one place. And this is based on, hundreds and thousands of compounds that Merck, Pfizer, AstraZeneca developed. They came up with these benchmarking to say, how are we going to move forward with our form and formulation? The Merck model, I call it, is the preclinical dose number, which gives you just the FASF solubility, gives you an understanding of how dose proportional your compound is going to be in any animal study. And what you also can have, well, you can start comparing different compounds, but there's also what's called a formulated preclinical dose number, where this here is just using straight up FASF solubility. But if you want to say use a cyclodextrin or some solubilizing agent, after you do a 2-stage dilution from SGF to FASF, you take that solubility, and we also have a formulated preclinical dose number. And then the developability classification system. And again, we're talking about parameter sensitivity here. And I know early on your clinical doses are going to be all over the map, but this will tell you when you're going to be in danger zones. With the predicted human permeability from the Keiko-2 using GastroPlus, and your potential dose levels and your solubility in FASF, getting an understanding at those dose levels, or compound versus compound, what your BCS DCS classification is, and when you're going to get into really some danger zones. I mean, if you're down here already, you got to go amorphous, right? Or prodrug, or find yourself another compound that's got better solubility, right? And it's pretty scary that, people will go through even phase 1 without getting dose numbers and without trying to see where they fit on this developability classification. And we can get fancy, start doing some MIMBA analysis, looking at a fraction absorbed. It's just a fancy representation of how your absorption drops off versus dissolution number, something that has to do with particle size. This will tell you, is milling really going to help me from a dissolution standpoint? Am I dissolution limited, and is milling really going to help me or not? And again, it's all parameter sensitivity based on what potential dose. You could look at different doses, like 10 milligram, 100 milligram, and say, okay, when's my oral absorption going to stop? And what's not shown here, we always also use a lot is a maximum absorbable dose. We'll give you an idea of using that form, how much is going to be absorbed in humans from a predictive standpoint. And this just goes through, which I've already been through. Well, I shouldn't have talked through all of them on the one slide, but here we go. This is mapped out here, and the equations are in here for you. And here we would expect dose proportionality at least up to 300 mg per kg. And something that I just realized, not realized, but it came to my mind again yesterday when I was talking to Fang Li, our formulator. Just because you're not dose proportional, you're still going to increase exposures, right? It's just that it's not going to be dose proportional. So it's not, it's not the end of the world that you're not dose proportional, but this will give you understanding when you get out of that dose proportionality. And it does a pretty good job. It's based on, I think when they had the article over 900 compounds, and I don't know if it's been updated since, but it's pretty good. So again, with the polymorph prediction, I'm not the expert here, but we partner with Biosimulitics. What I can say is they have novel means of addressing this, and we're really focusing on cutting down the cost and bringing it to the masses, and using it earlier on to really guide your developability. And so from a high level, what the output is from this is going to be predicted crystal forms with predicted energies and densities. Every dot here is a potential crystal form with a crystal structure, and you can get a predicted powder X-ray out of that. Now, the great thing about getting a single crystal very early on is that you can then from in the lab, you have your crystal structure. Well, where does it fit on this map? Well, if it's up here, you got some problems, right? Not saying that all of these are going to crystallize, right? But there's, you're pretty high up in energy. But and this happens a lot more frequently than not, you're down here, right? You're pretty sure that you've got your lowest energy form. And this really then minimizes the amount of screening. It's taken money out of my plate, but this is really the fastest way to do this, right? And the most efficient way to do this is combining these in silico techniques with minimal amounts of bench work. Now, the beauty of this working with Simulytics is you can get biosimulitics is you can get, since you have energy differences, you can get different solubilities, right? So maybe the difference in solubility here is 0.0002 milligrams, or 2 micrograms per mil, et cetera. And I mean, that might be a big deal, but if these energies are really close and the solubilities are really close, you're not even going to see this difference in your oral absorption. And how do I know that? Because I've built my PBPK model with GastroPlus. I put in the different solubilities and I see what impact it has to my oral absorption. And again, I should have just went to the next slide. But each of these will come out with a powder X-ray. And what we're working on as well is taking the powder X-ray from the lab and then clustering it here and doing pattern recognition to see if that's a little bit harder with preferred orientation. And early on, 5 milligrams of material, it's crystallized out, the crystals aren't that great. But you can do it, right? I mean, if there's peaks here, and what's nice is if say you have 5 medchem batches and one of them has an extra peak and you're like, oh, is that a polymorph or not a polymorph? Well, first you take a look at the DSC, see what's going on there, do a slurry experiment. But if you still are wondering about it, you can maybe take a look and say, oh, wait a second, I've got this guy here because I know my crystal structure, and this guy has that peak, and I'm seeing that peak. This is, you really need to start investigating that. So this just gives you an understanding really of the risks of your program. And when I talk data rich, people are like, well, that's a buzzword. it's not even a buzzword, but what do you mean by that, right? So what I mean is getting the most amount of information from every experiment, right? So when we're doing a polymorph screening and I call it a screening, but it could just be 2 vials, right? But you want to use every piece of information from that experiment. You just don't want to let it slurry there, take an X-ray and throw it away, right? No, like if it doesn't crystallize, put it at -20. If you get some solids, pull some out, get a PLM. You don't need to get an X-ray. Is it PLM? Is it crystalline? If it's crystalline, then yeah, maybe you not waste, but maybe you use 2 milligrams of material and get an X-ray. If it's not crystalline, let's split that in 2, right? And so now you've got one experiment that's turned into 6 experiments. Add some polymer in one of those to try to get it to crystallize, and the other one do a very slow evaporation to try to release supersaturation. It was already placed in -20 to drop supersat or to increase the supersaturation. But now we're going to try to slow evaporate it. You don't want to rapid evaporate it because that mostly likely will lead to oils or amorphous material. And then again, PLM, just get a quick microscope image. If it's crystalline, then get an X-ray. You don't have to go immediately with the X-ray. And also take into consideration the solvents. You don't want to, and this is where you use cosmologic. So I don't have to waste a 0.5 gram of material getting estimated solubility. I get one accurate solubility, ideally a water solubility, or ideally a FASF solubility. Then I go into cosmologic and say, okay, what's my approximate solubility in these solvents going to be? And then I want to use medium solubility here. Why medium? I want enough solubility so that I can get supersaturation, but I don't want to lose all my material, right? So if I get solids, I want them to be able to turn over. So you need at least 2 to 3 milligrams per mil solubility to get rapid turnover in a 24-hour time frame. But I don't want to lose all my material. And so that using the in silico can really help you towards that. So again, single crystal determination. We've got single crystal in-house, 10 to 15 milligrams of material. Someone with someone that knows how to crystallize stuff. They work their magic, and you get a crystal structure. And if the crystals aren't big enough, then you go to micro ED. If you still can't get it through micro ED, is it absolutely necessary at this stage? No, but maybe for like 60 to 70% of your compounds, you could get this rapid crystal structure for structure identification, understanding how the bonds are going together. If it eventually goes to a filing, you're going to have this in the filing, etc. So yeah, the case studies are in progress and I can't show them even if I had them. So at this point, I really like to thank you for your time and open it up to questions.
Yeah, thank you very much, Bob. Much appreciated. If anyone has questions, please raise a hand, post in the Q&A, come off microphone and ask. If no one has any questions, I guess I have a few for follow-up, but please do continue to post if you would like. I know much of large pharma will use powder and capsule bottle compounding for phase 1 in a quick to first in human phase 1 clinical trial strategy. Why do all of the front loading exercises, Bob?
Yeah, so that's a great question, Andy. And we're not saying that the compounding or simple powder in a bottle is bad for phase 1. That might be exactly what your target product profile is calling for and exactly what your clinical strategy is. But you're eventually going to have to formulate. And if you're going for an oral dosage, you're eventually going to have to know about your solid form, about solubility, about how you want to formulate that. And so this benchmarking really can take place. It'll work regardless of what your fastest formulation strategy is, and it'll give you a head start on your formulation when it actually has to occur. But it also simple compounding is not always simple. And so powder properties, the stuff can just float on water and stuff like that. And so understanding those properties with just small amounts of material are going to be very insightful, even for powder in a bottle or compounding. And give you a specific instance, what we currently have a client that's using OraBlend. They're planning on using OraBlend in their compounding for taste masking, but when we did the dose number calc, they're not going to get any exposures at the doses that they're looking for. And so we really need to know that, because now, okay, we have to do something in order to get the exposures you're looking for at the doses that you're looking for. So it's really trying to ensure a successful phase 1.
Excellent. So very technically and scientifically intriguing presentation today, Bob, and thank you very much for that. Wondering if you can sort of re-highlight the value proposition of levering this approach to get to phase 1. So in terms of time benefit, cost benefit, is there an approach particularly that you might recommend? And if so, can you parameterize that at all?
Excellent question. And one of the biggest, well, first and foremost, don't assume you always need a salt screening. And I mean, polymorph screening a little bit, yes, right? But I see people wasting a lot of money on formulation development early, right? So they just say, okay, I don't know what's going to work best. I'm just going to make a bunch of dispersions. I'll make some salts, make some freebase. I'll nanomill it. I'll throw it in a rat PK study. And then if that doesn't work, I'll do some more rat PK studies. I'll just keep doing rat studies until I find something that works that I could use for phase 1. And what we're saying is, like, hold the phone. Let's take a look. What's your dose number? Let's get some benchmarking here. Is a salt feasible? Like, maybe you're at that BCS or DCS4 where there's no way you're going to avoid having the amorphous. But that's also the beauty of the PK prediction as well, is that you get an understanding of, if my dose goes up, if my human dose goes up to 400, where am I going to be at? And what's the real advantages of, and this really minimizes, and this is, I talk about the 12% that voted against the animals early on, is that we can really, it's not, 7 weeks of doing GastroPlus modeling. We're talking, a couple of hours of putting in some parameters and saying, okay, let's not do that. Let's not do that dog study. Let's take some time and really find out the definitive PK study that's going to get you to your formulation for formulation for phase 1. And it's not always this long, tenuous journey. If you put the understanding in it early, you get on your path, you decide your path, but you don't do it alone, right? You bring in everyone involved in the program, because you don't want the formulator making the decision. You don't want the API person making the decision. You want everybody's input on what's best for your target product profile and your clinical strategy.
So you can reduce tons of sort of traditional benchtop formulation trial and error, then those discovery formulations, for lack of a better term, putting them into animal species, the time and money it takes to do that. You can do this through in silico modeling and simulation, much faster, much quicker for a much lower spend. And then,
and I mean, I'm okay with extra animal studies, but people don't, that, like, a lot of our clients don't understand the formulations that are going into it. And so
Right.
they're not getting the maximum amount of data out of it because, if you ask a question, well, how is it dosed? Well, we just had it in a method cell suspension. Well, what was the particle size? Well, we don't know. Well, was it ground? Was it milled? All these things kind of matter. And it's not, thousands of dollars. It's taking a PLM image, right? And, what's the approximate particle size? Are these like chunks or are they, small
Yeah.
particles? And
Yeah. So it can even be bad data on the back end.
right. It's just knowing what's going into what's your intent, what's your overall intent of the animal study, what's the material going in, and what's going to happen. You can predict, what's going to happen to it in the animal, and then you can validate those predictions with 1 or 2 animal studies. And then once you validated that, you have your animal model, and then you can, really build up your human PK predictions. And we're not eliminating animal studies altogether. What we're trying to do is have a smart development approach.
Sure. Yeah. Get better data, faster data out of those preclinical studies to better inform the clinical path forward.
Yep.
Cool. All right. Well, thank you, Bob. I don't see any other questions in the chat. Well, wait, maybe there is. There's a Q&A too, but maybe that was already addressed. That might be Ricky's question, which I responded to. So
Okay.
feel free to raise a hand. We'll do the 10-second silence countdown, play the Jeopardy music in your head, and if nothing happens, we'll sign off. And thank you all. The webinar will be available on the Simulations Plus website. Jasmine, did you have any final thoughts that you wanted to add or mention?
No, that was it. There was a question about a link to the recording. So I put it in the chat. If you just come back to our website, you can find the recording there. We'll also have the slides up. Yeah, and I invite everyone to follow us on social media. I put those links in the chat. It has been recorded for playback. Will be available on our website and YouTube. But yeah, that concludes our webinar for today. Thank you,
All right.
everyone.
Thanks, everyone.
Thanks, everyone. Thanks, Bob. Cheers.